Good afternoon. That was a heavy lunch. Butter chicken, pork, vegetables. So the best thing to do after having a big lunch is go underwater in a submarine. So this room, Community Assembly, Jenna, who you saw this morning, and I, Alex Smith, who you, if you haven't seen me, we're going to be the chairs for this Community Assembly, and we're going to, what's going to be our signal?
We're going to just do this, so we have 5 minutes left. We'll wait until you kind of look at us, and then you'll know that we've got 5 minutes left, and we should stay to ask questions.
And if you see this iPad in the front, Dirk Stanke from the Biodiversity Institute. Within the Twitter environment, we'll be throwing these up on Periscope. So if you are a Twitter person, you can alert your followers to get in the submarine with us and walk.
Nice. All right, well, we will begin the session. So we're starting the session with fizzing, Simeon fizzing.
So did you hear the words about the laser doesn't this works though, so you can use it to advance your slides.
Good afternoon, ladies and gentlemen, and you're all welcome to my talk. In this talk, I will be using DNA barcoding data to understand correlates of invasion success in Southern Africa. Invasive species are detrimental both ecologically and economically, and their negative impact in recipient environment has justified a renewed commitment to understanding why some species become invasive, whereas others fail to do so. In South Africa alone, the South African government spent an approximately $100 million between 1995 and 2000 on the Working for Water project to eradicate and control invasive species. These amounts are ill-afforded and are expected to increase in the future due to climate change and increased globalization. And as such, there has been a renewed commitment amongst invasion ecologists to understand what drives the invasion success of alien species. More specifically, there has been this key question to understand if there are any key universal predictors of invasion success. This is a very challenging question to answer, as correlates of invasion success requires a wealth of information on the species biology, which is usually not available for most species. And because of such data unavailability, some researchers have focused on certain clades or key families to try to provide broad generalizations. Again, new evidence is emerging that shows that correlates of invasion success differs as we move from one system to another. Traits that are strong correlates in one system might not necessarily be good correlates in another system. So consequently, we still lack a general consensus on key traits that correlate with invasion success. This now brings me to the aim of the study, which was to use DNA barcoding data to evaluate the ecological and evolutionary determinants of invasion success in Southern Africa, using trees and shrubs as our model species. To do this, we compiled a checklist of native and non-native species from literature. For the native species, we used the publication of Moraine et al. 2014 on savanna forest and the origin of underground forests, savanna fire and the origin of underground forests of Africa. For the non-native species, we used the publication of Henderson 2000 based on the Southern African Plant Invaders Atlas. We further supplemented this information with extra data from the comprehensive tree guides of Coit Paragraph on trees of Southern Africa. Using the checklist for over 6 years, we collected leaf material from different plant species across Southern Africa. And this project has seen huge success from our sponsors, most especially from our sponsors, where we had a huge logistic support from the Toyota Environment Outreach and Kigoko Mountain Reserve. It wasn't just about collecting species. We also had a bit of community engagement where we got the school pupils to teach them what it takes to conserve biodiversity. Using the checklist and the sequences generated, we constructed the phylogeny of the regional pool using as implemented in BEAS, using the core DNA barcoding region. We constrained the tree using 22 fossil calibration points, as opposed to the 28 fossil calibration points reported in Morin et al. This is because some runs failed to converge even after several hours of running time. Using the checklist, we collected a suite of putative traits that have been linked with invasion success elsewhere. We looked at traits on the pollination mode of the species, the flowering time, the dispersal mode, the seed mass, the maximum plant height of the species, and the sexual system. Oral statistical analysis were performed in R using the Picante, Kepa, and 5-code packages. First, we tested Darwin's naturalization hypothesis by calculating the phylogenetic distance separating each non-native species, that is the invasive and alien invasive and the alien non-invasive, from the nearest native species on the phylogeny. If invasive species are phylogenetically isolated from native species as predicted by Darwin's naturalization hypothesis, we observe that the average phylogenetic distance separating invasive from native species should be greater and it should differ from random expectation. We compared timing of flowering period by calculating phylogenetic ANOVA with post-hoc comparison of mean. We looked at when the species start flowering, the first month when the species start flowering, the last month when they stop flowering, and the duration of the flowering time. However, the coding of the flowering time could be biased, so we plotted a Fano curve to look at when most non-native species flower. It was somewhere around September. So we shifted the flowering time to September and coded September to be 1 and August to be 12. This was just to see if the way we coded the flowering time has a bias on our analysis. By calculating the phylogenetic independent contrast, we explored the relationship between invasiveness and maximum plant height, seed mass, sexual system, a dispersal mode, and a pollination mode using the branch algorithm. The phylogenetic independent contrast is a method that calculates species, that corrects for species phylogenetic non-independence while taking into account the trait difference between sister clades. The regional phylogeny is shown on the table with the invasive species in red, shows the distribution of the invasive species in red, the non-invasive species in green, and the native species in black. We found that the average phylogenetic distance separating the invasive from the native was significantly different to that separating the invasive from the non-invasive and from the native and non-invasive. However, this distance was not significantly different from the mean observed fell within our confidence interval. Surprisingly, we did not find any strong correlates of invasion success in Southern Africa, as opposed to other studies where these correlates have been found. But when we combine the invasive species and the non-invasive species to form the non-native group, we found that non-native species flower for longer period, and that difference was significant. Results were consistent irrespective of when the species start or stop flowering. Hence, our analysis were not biased by how we correct for the flowering time. For the sexual system, the pollination mode, and the dispersal mode, we calculated the number of potential contrasts with data and the number of contrasts differing in traits. And each time, the number of contrasts differing in traits were quite lower. And we found that only the dispersal mode and the sexual system were significant between the native and the native and the non-invasive, the non-native, sorry. For the discussion, we found no strong correlates of invasion success in Southern Africa between alien invasive and alien non-invasive, contrasting findings in other systems. For example, PSEC and colleagues in 2004, using temperate trees and shrubs, found that the invasive trees were taller, they flower earlier, and were biotically dispersed. Possible reasons for the non-significance in our results were we might have focused on the wrong traits, as new evidence is emerging that shows that the native range, the minimum generation times, are also key correlates in other regions. Or in this study, we focus on single traits, but there is also evidence that a combination of traits can increase the explanatory power. Or it might just reflect the context-dependent nature of invasion success. However, we found that in Southern Africa, non-native species that are more often abiotically dispersed are hermaphroditic and flower for longer periods might increase the chance of dispersal and establishment, but do not predict invasiveness. Both claims suggested by Darwin, that is, alien species that are phylogenetically isolated from native species to stand a greater chance of establishment, or alien species that are phylogenetically clustered to native should stand a greater chance of establishment. All these claims have reasonable explanation, and testing these claims has given rise to what's called the Darwin's naturalization conundrum. And even in some instances, with the same data set, the hypotheses have been accepted and rejected. But in this study, we suggest that these claims are not mutually exclusive, and we therefore, although we do not resolve the Darwin's naturalization conundrum, our results in the study might help move towards its resolution. And we just recently published a paper which was also chosen as the editor's choice. And I would like to thank the following for their support, most especially the SIT International Barcode of Life Committee for Travel Plan. Thank you.
We've got some time for questions.
I have a quick question. I was just wondering where you're going to go from here. I was just curious about sort of what communities are being invaded and the characteristics of those communities. Are you doing anything with sort of what's left over in these presumably disturbed sites?
For now, we don't really have such data, but I think the next thing is to move maybe towards getting some more threats and testing those ones that have been found that we didn't include. It's quite difficult to get the threats, but I mean, if we get some more threats, we can test to see if it actually explains.
Thank you. Next up, from Instituto Alexander von Humboldt in Colombia, Marilyn Gonzalez. Marilyn.
Lo siento. No worries. So, hi everyone. Well, I come from the Institute Alexander von Humboldt, and I will show you some preliminary results about a pilot that we have in the Pacific rainforest in Colombia. So, just to put you in context, Colombia is in the north part of South America, and is one of the most biodiverse countries in the world. And we used to say that Colombia is 50% forest, because we have the Amazonian rainforest, we have tropical rainforest, we have cloud forest on the mountains of the Andes, and we have an amazing rainforest here. Did you see something? No. It's not working. on the Pacific coast of the country that is also known as the Chocó biogeographic regions. So here I just put you some numbers with the diversity that we have in different taxonomic groups. And unfortunately, we also have a very high rate of annual deforestation. That is our mean annual rate is about 300,000 hectares that are lost every year. And the drivers of this deforestation is different in every region. But as we are focusing today on the Pacific coast, what is the main driver right now of tropical deforestation there is gold mining, illegal and legal gold mining. So those are current pictures of what is happening in this amazing biodiversity hotspot. So there is a RED initiative on this Pacific coast, and for those that are not familiar with that, RED talks about reducing emissions from deforestation and degradation. The plus is because these initiatives are now integrating like all the cultural heritage of the area, and the bio, it is because they are now willing to integrate other components of biodiversity. So stop seeing these forests just as carbon stock and start seeing them also as repository of a major biodiversity richness. So this particular RED project is led by BioRed, funded by USAID, and what they are doing is that they are screening all the Pacific coast with satellite images and radar images and trying to get some numbers on how much carbon it is getting stuck in those forests. And Humboldt is not at all involved in any RED project, but we saw an opportunity to get involved in this particular initiative from the bio perspective. So we propose to be part of this assessment of biodiversity in these plots. So in particular, they have to calibrate their carbon model. They had to establish some permanent plots, and we were working in 4 of them. And the 4 forests are here: Mutatán, Tepepe, La Plata, and Cajambre. And those territories are ruled either by indigenous communities, such as the Embera in the Mutatá part, the north part here, and the other ones are ruled by Afro-descendant communities in Colombia, so over here. So they have complete, let's say, independent rulement on the territory. And what we did as Humboldt was to get there and to assess different components of biodiversity. So we put camera traps, we put different zone meters in order to have an idea of the bioacoustic landscape. And in the permanent plots, we measure taxonomic, phylogenetic, and functional diversity of trees. I won't show you the results of the camera traps of the bioacoustic, but if you're interested, I can tell you more about that. Sorry, I thought it wasn't there. So our aim is to assess different components of biodiversity to see if we can add some conservation values to these forests. And doing so, we also want to see if these uncoupled patterns of diversity can help us to understand better the ecological mechanism that may be driving community assembly here. So now you know that there are several papers that have shown that patterns of different components of diversity may show uncoupled patterns, so they may be telling you different things about like the history of a place. So if you have an area that have few species, but in these few species you may have unique lineages, this is an area that is really important from an evolutionary perspective for conservation. And at the same time, these uncoupled patterns of diversity, when you are like over here and you know which is the phylogenetic relatedness of these species, or the distance relatedness in traits of these species, all the different assemblage of the local communities will telling us if this is like environmental filtering driver communities, or if those are communities that are driven by competition. And so there have been in the last decade several works in tropical trees, and they have shown like different results. For some of them, we have found that environmental filtering is really important. In other cases, competition has been like the major driver of community assemblage. But everything here depends on your geographic, taxonomical scale, and also the methodology that you use. So what we did was to go to these 4 places, and we sampled the trees with a DBH higher than 10 centimeters. We collected, for the voucher to the herbarium, the tissue in silica, and we record different measures of functional traits that I will explain to you just later. So those are some results from the taxonomic diversity. So you see that the 3 first places have like more or less similar number of individuals and more or less similar number of species, up to 180. But the last place, Cahambre, has like more than 1,000 individuals in only 21 species. And this is a flooded forest. It is dominated by these 2 species, a palm and there is a legume. So those are like really far in the phylogeny, but these may have like 500 individuals and this one 600. And the remaining species are just shared between the other kind of trees. So really contrasting plots. And of course, when you do like some indices of diversity, what you find is if you are not accounting for abundances, Mutata will be the plot with the highest diversity, whereas Cahambre will be the one with the lowest diversity. But once that you account for those evenness in the diversity, well, both the 3 first forests will be like quite similar in the range of diversity. So for phylogenetic diversity, what we did was that we were able to sequence some of the species for RBCLA and tRNAL, and of course our goal is to have all our species sequences with these all 4 barcodes, but for this exercise today I have to assemble like super matrix with the sequence that we have so far and some sequence that we recover from GenBank. So with that we also built a phylogeny for all the species that were present in the 4 forest plots, and we had 3 measures of phylogenetic diversity. One was phylogenetic diversity per se in the census of fate, so it's the sum of the branch lengths that we corrected, let's say, by the number of species present. We have also the mean pairwise distance of phylogenetic diversity, and what we do is that we have our observed value that we compare to a random distribution of value. So what we are looking here is that if our communities have species that are more closely related than if they were randomly sampled, in that case we will be having a phylogenetic clustering. But if our local community has a species that are more distantly related than randomly sampled, then we will have a phylogenetic overdispersion. I hope this is more or less clear. I'm sorry for the question. I'm sure you're understanding everything. Okay, so some of the results with the super matrix phylogenetic diversity, what we found is that 2 of the communities, PDPP and Cahambre, apparently have less phylogenetic diversity that they should have if they were just randomly sampled. But that doesn't account again for the abundances. Once that you put abundances in the equation, so here in the NRI index, the pattern just invert a little bit. So this is not significant. This means that all those plots are assembled like in a random way from the global phylogeny, but you can see that the tendency is to be over-dispersed. And something that was interesting just preparing this presentation was that I did an analysis putting just one species, that was Samia roesley. I don't know if you're familiar with that, but Samia is a gymnosperm. Samia is a gymnosperm, so it's completely far away from angiosperm, that are the main major group in our study. And when you just put the presence of the species, everything changed, because Cahambre just like shoots its phylogenetic diversity. So Cahambre was this place where you have a dominant palm, a dominant legume, and in the understory, you have a dominant Samian. So if you put the dominant Samian there, Cahambre have like a huge amount of phylogenetic diversity. We did a measure of evolutionary distinctiveness, and so what data count for is like how long is the branch length and how many descendant nodes do you have from that branch length. So you are more unique if you have a long branch length and you don't have any sister species. More or less that will be that. And of course, in this case, Samia will be like the species with the highest evolutionary distinctiveness. But we have other cases. And for instance, this Asteria, it is the only representative of the family in the dataset. It is an Olacaceae. This Orphanodendron vernali is the only species of that genus that is known and is local to the Chocobio region. And this Magnolia catorium, I put you here just a description that is in the IUCN Red List. So this Magnolia catorium is currently only known from a single locality, which has been highly degraded. And in fact, Magnolia comes from the basal lineage of Angiosperm, but it's also critically endangered. So it has like two major values for conservation here. If you lose that species, you are not only losing a species, but also a huge phylogenetic history. We also made some measures for functional diversity. So we measure wood density, dry contact leaf matter, and specific leaf area. Of course, we try to follow all the protocols that exist for functional diversity, but most of them were prepared in temperate forests, so they tell you that you have to have leaves that are fully exposed in the sun and that don't have any epiphytes or parasites or anything, but when you work in tropical forests, that is just impossible to have a leaf that is a proper leaf. So we spend a lot of time like cleaning the leaf just before having the weight and everything, but yeah, we didn't do it for all of them. We have the density, dry contact leaf area, and specific leaf area. Those are traits that are related with the resource acquisition strategies. And we analyze that in a similar way as the phylogenetic diversity. So we create a distance matrix, and we were just looking at the dispersion of those traits. And what we see here is it is below the zero, so, but nothing is significant. So it seems that the communities are assembled in a functional way where they look like randomly assembled. So, but they tend to be clustered. So in summary, what we found was random patterns of either phylogenetic and functional diversity, but phylogenetic diversity had a tendency to over-dispersion, whereas functional diversity had a tendency to clustering. So this is something similar to what has found by Enquist and Swenson in the tropical dry forest of Guanacaste, and their conclusion there was that you have opposing mechanisms either from environmental filtering and competition that are acting, and so the signal that you get in some cases is like neutral dispersion. And well, of course, DNA barcoding is allowing us to improve the taxonomic knowledge of the sample trees, and it has been really useful as a baseline to reveal phylogenetic phylogenetic hypothesis in order to assess phylogenetic diversity. Now, I think that it's really important not to forget in those cases also maybe to start integrating other components of the forest, not only the trees, because in the understory we have really interesting things that can change what we are seeing there. And of course, I really think that all those threats projects have to stop seeing the forest as just carbon stock and start integrating other components of diversity. So thank you to the USAID BioRed program, the Ministry of Environment, and the CDB through the Japan Biodiversity Fund sponsored my trip to here. And thanks to all of you and the people from the community that allow us to work there. Thank you.
We probably have time for a quick question if there's just one.
How do you get your functional diversity on wood density area? How do you get to the functional diversity?
You have different ways to measure that, but what we did there is to create just like a distance matrix from all the different traits, And then you can analyze that in different ways. You can make like, let's say, a cladogram. So you will make a cladogram of distances. And you have the index of Pesci and Gaston will be like the equivalent of the PD for phylogenetic diversity. So only the sum of the branch lengths. But with that distance matrix, you can also make like mean pairwise distance of traits or mean nearest neighbor distance of traits. So the only thing that you really need is this distance matrix of traits, and I can show you some of them later. The way to do it.
Thank you. All right. All right, and next we have Brandon Mong-Boji from the University of Malaya.
Hi everyone, my name is Brandon Mo from University of Malaya, and today I'm going to present about the optimizing meta-barcoding of tropical Malay strap samples and preliminary results on seasonal changes in insect diversity in peninsular Malaysia. Have you heard about extinction? How about mass extinction? 5 mass extinctions have happened on Earth. The first 2 were due to global cooling. 3rd and 4th were due to global warming. The 5th one hasn't been determined yet. It might be due to asteroids. And right now, we are entering the 6th mass extinction. For real? Yes, unfortunately, we are entering the 6th mass extinction. Mass extinction doesn't just involve big mammals, but also involve small invertebrates, for example, insects, which are the majority of life on Earth. Global temperature has been increasing since 135 years ago. The first ever record has been recorded, and the temperature is still keep on increasing. And for your information, the year 2014 was the warmest year on record. An article suggested that we are entering the 6th mass extinction, and one of the reasons is due to climate change. And there's another review article suggested that world biomes are vulnerable to climate change. Tropical species were thought to be at lower risk to climate-related extinction. However, as you can see from this figure, the concentration of tropical species which are vulnerable to climate change is quite high, especially at the equator and at Southeast Asia. Few taxa has been studied for their vulnerability to climate change, and as for your information, the number of studies on insects is quite low. Insects are ectothermic, which means they are affected by temperature. For example, insects in temperate region, for example, those in Canada, which exhibit seasonal activity, experience a very wide range of temperature, around 30 degrees Celsius of difference. However, tropical insects, for example, those in Malaysia, which thought to be aseasonal, experience a very narrow range of temperature. It's around 2 degrees Celsius of difference. Thus, physiologically, tropical insects might not able to cope with slight increase of temperature. Thus, in future, if temperature keep on increasing, tropical insects might experience a high temperature which has never been experienced before, and this might lead them to heat shock and death. And due to cascade effects, it might affect other higher taxa, and it will lead to mass extinctions. And here comes the 6th mass extinctions. You die, I die, everybody dies. Alright? Alright, for my objectives is to investigate patterns of seasonal changes in activity of insect species across Peninsular Malaysia, to determine the extent to which insect species in Peninsular Malaysia exhibit seasonal activity. And the last one, to determine if different ecotypes show different types of patterns. So for the methods, we're going to use Malay strap meta-barcoding. We use Malay strap to collect the insects, and we have going to collect continuously for 1 year, which means 12 months. And we're going to use method barcoding to determine their species. We use method barcode because it's cost effective and efficient compared to the conventional way. So for Malay strap method barcoding, we're going to collect the insects in a weekly basis, every Friday. After that, I'm going to sort the insects out, bulk DNA extraction. After that, proceed with bulk PCR using a universal primer set, which amplifies all the CO1 gene of insects. Send the amplicons for high-throughput sequencing. The sequencer that we're going to use is Illumina My6. The last one, data analysis. Okay. So before we start our main study, we have conducted our pilot study. Method barcoding is a new study. The major steps that will influence or affect method barcoding is bulk PCR, sequencing, and also data analysis. That's why we're going to study them. Okay, for bulk PCR, as I mentioned before, we need to establish an optimal primer set, a universal primer set which can amplify all the CO1 gene of the insects. Thus, in this pilot study, we have tested 5 different types of universal primers, and this is the results. Objective is to establish the optimal primer set for method barcoding of Malay-strap samples, and also to establish an optimal biogenic pipeline. Alright, so we have 80 different types of insects, different species. We have collected, and their DNA has been extracted individually and amplified individually using 5 different types of primer sets. And this is the result. Alright, so this is the result. So 5 different primer sets. The result is the red primers and the green primers showed a very good result. It's around 89% of detection rate. That's why we didn't continue for their studies. After that, as I mentioned before, 80 species we have extracted their DNA individually. Right now, we're going to pull off all of the DNA together, and we're going to amplify them in one go simultaneously using that 2 types of primers separately. And for the result of bulk PCR, the green primers showed a better detection rate. It's around 90% of the detection rate out of 80 species, and the red is, and for the red primer, it's around 80% of the detection rate. Next will be the data analysis. We use 2 types of pipelines. The first one is automated pipeline, and the next one is manual pipeline. For the manual pipeline, we use codon code, aligner, and also bioedit. It's a very easy to use pipeline. For the automated pipeline, we use around 3 to 4 hours to process 1 file, while for the manual pipeline, we use around 6 to 8 hours just to process 1 file. Alright, so for the manual pipeline and the automated pipeline, this is the result. After the filtering and also quality control pipelines, the manual pipeline showed a better result. As you know, we have 80 species, right? So we should get around 80 species, 80 OTUs, and the manual pipeline showed a lesser OTUs compared to automated pipeline. However, the analysis were complicated with some unexpected OTU. So what are the unexpected OTUs? It might be due to contamination, chimera formation, mitochondrial heteroplasmy, and also pneumite. To compare our pipelines to other published pipelines, our pipelines showed acceptable detection rate is around 80 to 90%. So for the conclusion for my pilot study, the single universal primer sets used here showed an acceptable detection rate around 80 to 90%. And for the green primer set will be used for the main project. For the main project is to determine the seasonal activity of the insects, tropical insects. Alright, as you can see, 6 months of insects has been collected, and the number of individuals has been calculated, and this graph was plotted against temperature, and also precipitation, rainfall. And from this graph, you can see when when the temperature increases and also the precipitation increases, the number of individuals decreases. And I have three types of ecotypes. The first one is botanical garden. The second one is oil palm plantation. The third one is a forest reserve. So this is oil palm plantation. As you can see, there's one missing data here. It's because one of our Malay stripes were being stolen. Okay. Yeah. So this graph also shows the same pattern. Thank you. And this project was funded by University of Malaya High Impact Research collaboration from Monash University. And I would like to thank my helpers who has assisted in our work. And I would thank British Council and also other institutions which has made this presentation possible. Thank you.
We have time for questions. Way back there.
Yeah, what was the read length on that the primer that you selected?
Read length is around 313 base pair.
Okay.
I have a question. The other, if I saw the primer names properly,
Okay.
the two regions that you were looking at were not really overlapping. They were one at the 5' and one at the 3'. Is that correct?
Yeah. Okay.
So that's good. All right. Yeah.
Yeah. They don't overlap. Yeah. They don't overlap.
So the first pair used mini left arm, ML
Yeah,
left arm
yes.
one. Is that so? What you missed probably was Hymenoptera. Does that match what you remember of your results? Because that primer doesn't work well with Hymenoptera.
Yeah, we have some primer bias over there.
Yeah.
Yeah. Hymenoptera has a lower detection rate.
Yeah. Okay. Cool. Is that a question or a filming? Okay. Drop back to the schedule. And Gillian Martin, ex of the University of Guelph, currently of the University of Quebec in Montreal, is going to be talking about her work.
So as Alex said, I finished up my master's in Guelph 2 years ago? I can't remember. And now I'm moved over to Quebec to do some work there. And it all kind of ties together about metacommunity structure and DNA barcoding. So it's going to be a little bit about, mostly about my master's, and if I have time, a little bit about my PhD because it just started in September. So, all right. So the community structure has been a long-standing area of debate in community colleges. No one really knows what the processes are about behind it, and there's a lot of theories about what's actually at work. So the two main theories are niche theory and neutral theory. And a niche is the range of ecological requirements that a species needs to live and reproduce. And so you would expect that as species interact with other species and other individuals of those species, their niche will shrink to become their realized niche. And in the diagram on the left, you can see the niches are in different colors, and then the species always match in their same niche. So species 4 is always found in their orange niche. However, some people find that niche theory doesn't adequately explain the high levels of diversity found in areas such as the tropics, where there are lots of species within what seems to be exactly the same niche. So instead, neutral theory proposes that species are exactly the same in terms of birth rates, death rates, speciation rates, and dispersal rates. And instead of species matching with their specific niche, instead they're found randomly dispersed throughout the environment based on whichever area they populate first. However, while niche theory and neutral theory kind of explain stuff, and they have worked for quite a bit of stuff, they don't really take into account the evolutionary history or regional processes like dispersal. So metacommunity theory was developed as a way to kind of correct that. And it incorporates both components from niche theory and neutral theory. So you would expect that it adds it adds in dispersal ability to take into account the regional processes that might be at work in an area. So as your dispersal ability increases, you expect that the
role of local processes like environmental variation would decrease because species are no longer restricted by connectivity or their ability to get from site to site. So it's been tested in a variety of ecosystems, from bats to plants to invertebrates. And it's been found to work, but there hasn't really been a consistent answer. No one really knows where along that line everything should fall, and it's kind of been mixed. Some fall closer to the top, and some fall closer to the bottom. So what we do know is that species do have specific ecological preferences, and they do have certain environmental tolerances. Sorry. You expect that as species community composition changes, you'd expect that would reflect the changes in the environment that are happening. So as species niche gets narrower and narrower, you would expect that would reflect smaller and smaller changes in the environment. But by ignoring taxonomic resolution in meta-community ecology work, you don't really know whether or not the process that you're detecting would be consistent throughout different regions and different data sets. So as you can see in the little trees that I have, if you look at two different communities and you look at them at a species level versus a family level, you could have very different results between sites. And meta-community theory hasn't really addressed this idea of the differences in taxonomic resolution. But with the advancement of DNA barcoding, it's a lot easier to add in that additional information to answer these questions. So I had two different objectives. I was looking at whether or not there's a relationship between taxonomic resolution and meta-community model explanatory power. And I had two different hypotheses. So the first was that if you group species together, you would decrease the explanatory power of your model. And this would be because by grouping species, you're masking the relationship between environment and community. Alternatively, grouping species could increase the explanatory power of your model, and this would be because you'd be revealing the relationship between community and environment. So species are more similar than families are. Right. So my second question was to test whether or not that relationship is consistent across a variety of taxa. And to do that, I went to Algonquin Provincial Park, which is about 4 hours north of Guelph, and I sampled 21 different stream aquatic insect communities, and they're indicated by the red circles. And as you can see, there's a little bit of spatial bias. Everything is focused mainly around the Highway 60 corridor on the bottom, and that's just because it's a lot harder to get off around to the north side, and so hopefully by incorporating the spatial data into my analysis, I'd be able to detect whether or not there was an impact. So at each site, I followed a standardized biomonitoring protocol to sample the aquatic insect communities. I selected 3 different transects, so it would be 2 riffle transects and 1 pool. And I used 2 riffles because the riffles have been found to include about 80% of the species found, whereas pools are much less diverse. And at each transect, I conducted 2 stationary 1 meter squared kicks for 2 minutes, disturbing the sediment and collecting it in a net downstream. And then from each transect, I would pick out a minimum of 100 specimens from each transect, and then pool that to create 300 specimens total, at least. And then, in addition to that, I also characterized the environment at each site, and that involved sampling water chemistry, measuring the types of detritus present, or the substrate types as well. And I then used the environmental data and the spatial data to create an environment by site matrix and a site spatial matrix. And so then I was left with all these invertebrates to deal with. So instead of me identifying all them down to species, because I'm not a taxonomist, and it would have been really hard to reach that species resolution for myself, I used DNA barcoding to delineate my species units for analysis. And I used standardized protocols developed here at Bio, so I had really high sequencing success, particularly because I had insect order and very fresh tissue. So because I had so many invertebrates, I focused on 6 groups of taxa. I focused on the Coleoptera, the Diptera, the Ephemeroptera, the Olichera, the Plecoptera, and the Trichoptera. And I picked these because they were the most abundant in my sites. And so once I had that, I had to create my site by taxa matrices, and I created them at 3 different levels of taxonomic resolution. So I use the bin, species surrogate at a genus level and then at a family level. And to reach those counts, I randomly selected 20 specimens per family per site for each of the groups that I found. And I then sequenced those to reach my IDs, and I also morphologically ID'd them down to family and genus whenever needed. However, due to limitations of the bold reference library, I was unable to reach a genus level data set for the Diptera and a family or genus level data set for the oligochaetes. So with those three data sets, I created 3 different site-by-taxa matrices at the 3 different levels of taxa resolution. And I used variation decomposition to determine the relative roles environment and space played in structuring these 6 different communities. So my observations were that as explanatory power increases as the resolution becomes more coarse. So to explain this graph, along the x-axis is the mean percent divergence within taxa, and I calculated this using just basic K2P methods, and I did that because a family level in one order may be very different from a family level at another order, and same with the species level identifications. Hopefully by using the genetics, it would be more comparable across taxa than if I had relied on just taxonomic morphological identifications. And then so as you move from the left to the right, you go from fine resolution to coarse resolution. So on the left-hand side would be my bins, so my species surrogates, and then on the right are my family level groups. And then along the y-axis is the percent variation explained by the environment. I did run the analysis for space, but space was not significant for most groups. It was only important at a species level for the Plecoptera and the oligochaetes, so it wasn't as important in structuring those communities. All right, so as you can see, there's quite a bit of variation in the different groups, but there is a strong, significant upward trend. And you can see that the purple, the Ephemeroptera, had the strongest relationship, and they had about a moderate species richness. Oh, okay. And the orange line is the Coleoptera, and they had the weakest relationship, but they also had the least amount of diversity. So I probably just didn't have enough data to get a good result from that. So was there a relationship between taxonomic resolution and meta-community model explanatory power? Yes. As I decreased taxonomic resolution, I increased the amount of variation explained in my communities. And this relationship was not consistent across taxa. And this is likely because of ecological variations between the groups that I wasn't able to look at further. So in conclusion, comparisons across different taxonomic levels will have a really significant impact on your analyses. So if you're running a community analysis and one data set is only identified to, say, genus, and one data set is identified to species, you could be comparing very different results. So you could have a value way up there for your coarser levels, and then way down on the left for your species level. And this is a problem because in a lot of community studies, there is a mix of taxonomic resolution, especially for groups that are really hard to ID or just take a very long time. And really quickly, for my PhD, I'm taking this a step further, and I'm going to look at how habitat productivity affects species and genetic diversity. I'm also going to look at how species similarity, so functional redundancy and cryptic speciation affects community structure, and the relationship between species genetic diversity and meta-community structure. And to do this, I'm using a data set of 325 zooplankton-like communities in 5 different regions across Quebec and Canada. And they've been morphologically ID'd already, and I have about 62 species to look at. So in the future, you'll probably hear about some of the work I've done from that. So with that, I'd just like to thank everyone who helped me here as well, and thank you for listening, and I'll take any questions. Yeah.
Hi, have you ever tried oligotyping on your data set?
Sorry?
Oligotyping?
I don't know what that is.
So sub-OTU, there's variation inside the OTU, right?
Yeah.
And so, what's his name? Murat Eren, E-R-E-N,
Okay.
has some software, oligotyping, and then something called minimum entropy decomposition, so MED,
Okay.
right? Will break down OTUs into sub-OTUs, and down to one nucleotide difference. And sometimes you see differences, say, in which OTU or which sub-OTU is present in which season. So a really nice example of that, which is not from Aaron, is the Nolte et al. Paper in molecular ecology a few years ago. It was one of the first marketing papers.
Yeah.
And it's a classic. It's hardly cited, but it really is, I think, one of the most important ones. So Nolte et al. In the Schlatter lab, the later type of It'll be really nice because that data set you're going to create, right?
Yeah.
Is perfect for that.
Yeah, I am doing next-gen sequencing for that data set, so I've read some of the literature, so that sounds vaguely familiar. But I will definitely look into it further. Thanks. Any further questions? Yeah.
Do you know in as much your study site was influenced by luck events? Because normally in aquatic insects there is a strong local movement, so some escape via drift, and the other go to stable stones, which could influence your data set.
Yes, so I don't know about flood events specifically, but I did return to Algonquin the following spring and summer, and I did notice that a couple of my sites had dried up by the time I got there in the spring, so there could be some variation temporally over time. But it would be interesting. It would have been interesting to include that to see if there was an effect. Yeah, okay, real quick.
Sorry, because I wondered if you tested about what in general, what intelligence are useful for the assessment of water quality. Have you tried to compare some index, to compare it level of resolution?
Whether or not the water quality is so no, but Algonquin is pretty much a pristine environment, so there shouldn't have been too much impacting that. That is one thing that would have been interesting, would be compare whether or not this is consistent between sites or between areas that are impacted to reference condition sites, which are the ones I use. So no. Okay, thank you.
And our last speaker of this section is Catherine Abbott from Fisheries and Oceans Canada. Right there. Pointer does not work. The advance is looking a little shaky these days, but if you just do that, it'll advance.
And then otherwise,
That's good, sure.
Okay. Okay, thank you. Okay, great. Thanks for staying. I don't know if anyone who's standing wants to grab a seat, but there are a lot of seats here if you feel like it. I'm going to talk to you today about an integrated approach that links high throughput taxonomy with community ecology. The motivation for doing this work is funded by Canada's federal government genomics initiative called the Genomics Research and Development Initiative. We have an interdepartmental project on quarantine and invasive species where multiple departments are working on generating high-quality sequence reference libraries that can be used, multi-sequence, high-quality, that could be used in a regulatory context for identifications of regulated invasive species. So I have a fantastic postdoc, Magali Castellan, who is working on building this library and essentially conducting a molecular inventory on the coast of BC. And she's developed these methods so that the library has robust species delimitation methods used, and she's established this way of linking this large kind of biodiversity data set with community ecology. So Magali can't be here because she's presenting at the American Fisheries Society meeting this week. So human disturbances like overharvesting, urbanization, pollution, invasive species, and global environmental change all generate selection pressures that affect the distribution, ecology, and evolution of species, and this in turn can of course affect community dynamics, ecosystem functioning, and indeed global processes. Species responses to human disturbances are often poorly understood, and this is largely because community dynamics are based on complex and often unresolved ecological networks where each species that's interacting in the community has its own life history traits and inherited genetic complement and competitive abilities within the community. So there's some evidence that focusing on a set of interacting species and elucidating their ecological relationships, the nature and strength of these relationships can improve conservation strategies.
An example of this is from Wilmers et al. In 2012, who worked in the Northeast Pacific and found that sea urchin populations were suppressed in coastal marine environments where sea otters are present. And indeed, where sea otters are present, there tends to be well-established kelp forests that were very effective at locally storing atmospheric carbon. So in this context, this is obviously oversimplified, but if you had a conservation goal aimed at reducing your carbon footprint, one strategy would be to protect sea otters and retain the kelp forests. Again, it is oversimplified, but it provides a little bit of an example of how focusing on a set of interacting species and understanding their ecological relationships, the dynamics that are at play in the community can directly lead to operational guidelines for management. So we can test species co-occurrence patterns using ecological null models, where the null hypothesis is that species are distributed according to random processes. And this can provide insights on community dynamics, including on the nature and strength of ecological relationships. In the case of the sea otters, if this approach were applied, we'd find that kelp and sea otters co-occur more often than expected by chance, and that each of the kelp and the sea otters co-occur with sea urchins to a lesser degree than expected by chance. A benefit of this type of approach is that it only requires presence-absence species data. So these are the kinds of data that we're getting in our biodiversity assessments when we're barcoding and doing molecular inventory. And so it could provide a really good tool for investigating species interactions at large taxonomic scales. However, obtaining realistic scenarios out of this approach requires relatively complete and accurate species lists where species are defined using some conceptual framework of evolution so that they are actually reflective of distinct lineages that incorporates their evolutionary history. And indeed, inaccurate species-level taxonomies can confound ecological studies and here could induce misleading management strategies. So of course, coalescent-based approaches provide a really good means for species delimitation, where they model speciation and coalescent processes on a time-calibrated tree. For example, the GMYC method that's commonly used, developed by Pons et al. A drawback of these methods is that they're computationally intensive and they're suited to taxonomically narrow data sets. So it's impossible to apply them on a whole, biodiversity data set where you actually want to analyze all of the species that you've collected. So this generates a need for pragmatic tools and techniques for delimiting species in large biodiversity data sets. So our objective was to find a novel synthetic approach to reconciling taxonomy and community ecology. We wanted to enable fast and robust biodiversity assessment across multiple ecological communities and to be able to identify primary factors affecting the distributional patterns of communities. We used the British Columbia coast as our study area because it's one of the most productive environments in North America. It contains a variety of seashore habitat types, including kelp forests, rocky reefs, mudflats, sandy beaches, and a gradient of human disturbance where the Strait of Georgia at the south is actually considered by Parks Canada to be the most at-risk natural environment in Canada. That was news to me. So among marine macroorganisms on the BC coast, shelled gastropods and brachyuran crabs are two of the most diversified taxon groups. They're also relatively well studied, and this means that there's a fair bit of information available on their distributions, ecology, and general biology. And they're also a good model here for studying species interactions because they have a long coevolutionary history as predator and prey. So what we did is we sampled gastropods and crabs, brachyuran crabs, intertidally and subtidally down to a 10-meter depth at 52 sites across 4 marine ecosections around Vancouver Island and up the south coast of British Columbia. In total, we collected 1,000 individuals, more than 1,000 specimens, and they were initially sorted to superfamily using morphology, and then a subset, about 50% of those specimens, the 50% picked to incorporate, to cover the morphological variation that we saw. They were selected for molecular analysis with CO1. We queried bold and imported records that matched our study taxonomically and geographically. And then we, for each superfamily, we plotted pairwise CO1 distances to check for multimodal versus unimodal distributions. For superfamilies that had multimodal distributions, we applied two phylogenetic methods side by side to compare them and to generate what we refer to as primary species hypotheses. So we compared the GMYC method with a newer PTP method, which doesn't require the generation of a time-calibrated automated tree, and so it's a lot computationally faster, and so it may provide an easier and equivalent, equally effective method for developing primary species hypotheses using CO1 as the popular GMYC. After that step, we used an integrated approach to taxonomy where we applied independent taxonomic characters, namely morphology and 28S sequences. And we looked at those partitions and developed robust species hypotheses that were then assigned a valid name based on available literature and databases. The last step was we identified morphological characters that were diagnostic of these robust species hypotheses, and then we applied those morphological characters back to the specimen pool that hadn't been sequenced. So we ended up with an entire collection that had been was assigned a name, the name existed, and it was essentially identified. So we did this approach on 19 superfamilies, 11 of which required phylogenetic analysis because more than one species was sampled within a superfamily. In total, we analyzed 91 species, 57 of which were sampled within our collection. And we found that the PTP phylogenetic method was very congruent with the GMYC. PTP is shown always on your left. And in only two cases they were discrepant. We know that GMYC is sensitive to sampling biases and small genetic distances, and it can be a little bit prone to oversplitting. In the two cases where GMYC and PTP differed, that's indeed what we saw. How we resolved this was first we looked to the independent taxonomic characters that we were analyzing in the study. So we looked to morphology and 28S. And in the case of the Buxonoidea example, morphology and 28S agreed with each other and with the primary species hypothesis generated by PTP, and so that pretty much resolved it. But in the case of Trichoidea, we didn't have 28S sequences because these data came from Bold. And so we turned to inevitably more subjective data types, including we looked at this split in the context of the range of interest-specific genetic distances that were observed elsewhere within that superfamily, and we also looked at what was known about life history traits and geographic distributions. So in this case, it's known that this family has only walkaway larvae, they're not long-distance dispersers, and they're known for high levels of population structuring. So that was the decision taken here. Okay, so what we did is once the species were delineated, we used common descriptive and inferential statistics to look at. We incorporated the geographic sampling data, and we looked at the distributional patterns of communities. And we also looked at species co-occurrence patterns by testing them against null models to see if we could find a causal basis for the observed patterns. So a few results. We found that species richness and evenness were higher on the west coast of Vancouver Island compared to the Strait of Georgia, and that this is consistent with the west coast being open shore environments with lots of upwelling and well-oxygenated water, therefore supporting high productivity environments. Whereas Strait of Georgia operates more like an inland sea. It had large sheltered estuarine and marine mudflats that were characterized by few but highly abundant species. We also found a high proportion of rare species in the Strait of Georgia. This patchiness, we think, is driven by habitat heterogeneity. Certainly there's some rocky habitats characterized by small islands and rapid tidal passages that have a unique complement of species. And there's also higher urban activity in this area, ferries and marine and areas that we thought were also affecting the patchiness. So it's important to know about patchiness for biodiversity studies because, of course, if the goal is to sample everything that's there, this patchiness translated into requiring, I think it was about a 6-fold higher sampling effort in the Strait of Georgia compared to the west coast of Vancouver Island to achieve this to actually collect the same kind of proportion of species that were there. And so since species numbers are often used to promote conservation interests, it's important to consider patchiness, in this case in the Strait of Georgia, and increase the sampling effort accordingly. So we also found that considering the whole species assemblage sample, that species co-occurrence patterns deviated from what we expected by chance, and that this was driven by non-random association of grazing gastropods. We think that this might be because grazing gastropods, compared to crabs and carnivorous gastropods, they're lower. They occupy a lower level, a lower trophic level, lower in the food chain. And we think perhaps, this is just one hypothesis, but that they might be ecologically more constrained and so subject to more pressures and more constraints, meaning they would be subject to bottom-up pressures that are physical effects that are acting on the food resource, and top-down pressures including grazing competition and predation. And so they may have less flexibility and be therefore more vulnerable to random colonization and extinction dynamics. And then the last thing to leave you with is that, interestingly, the highest number of co-occurring associated species were the grazing limpets, and they were also the most difficult to identify the species level. So this is where the molecular approach was particularly handy. Lots of people to thank, and the Canadian Aquatic Invasive Species Network for their collaboration. Thank you.
There is a question.
How do you use Bayesian PTP regularly?
Say it again.
Use the Bayesian PTP?
So Magali did this. Is that newer?
The newer one.
Yeah, we didn't use the newer.
I'm learning to hate it.
Are you? You know what? Magali's been trying to use it on a different data set because of some reviewer comments, and I don't think she liked it. Or I think she was, not that she didn't like it. I shouldn't say that in front of all you guys. But whatever you just said, she was, I don't know what the struggles were. This is not the new one.
And then use these two or these one for the GMOC?
I don't know. I can let you know. Like, I'll have it in the draft. She's going to submit this paper really soon, so it should be out soon. But those, yeah, I can.
Beast is very well named, right? It's a beast.
Yeah.
We're trying to figure out how to best prevent it.
I wish I could tell you right away, but I can tell you soon.
I have just a small curiosity. You used Cohen sequences, and you used both as an ID engine, right? But you didn't store the data.
Well, we used bold sequences, but I mean the species delimitation and identification was part of our pipeline. We didn't rely on bold for it.
So you didn't upload your own data to bold. That was my question.
So the data, because this is part of this Canadian Government Genomics Initiative, part of that initiative is to generate a Government of Canada-wide sequence database that can be used for regulatory applications. And the little key thing there is that certain departments deal with sensitive data that can't be released. So the point of this is to have a repository that's really high quality and highly traceable, that's multi-gene, and that's accessible to government scientists even before all data can be published. Okay. So I'm explaining all that because of the nature of the work, we've streamlined all our data into that federal government database, and from there it will get uploaded to BOLD, like automatically, magic. You know, the bioinformatics people hit a button and it's all done for us. And it hasn't happened yet, but we think it's really close. So there's going to be a kind of a glut. These data will definitely end up public on BOLD.
Yeah, I think it will be very useful.
I know, and it's a little late. I feel a bit bad about that, but what can you do, right? Life happens.
Should I switch off?
So,
correct me if I'm wrong, but I mean, you've basically gone through a cycle of getting the molecular information from closed eyes specimens, using the algorithms to cluster, get OTUs, do morphological identification based on those OTUs back to the original specimens.
The morphological assessment was independent. Like when we used the morphology in the integrated approach, the morphology was done independently of the phylogenetics. Yeah, yeah, and then that's right. And obviously, same with the nuclear 28S is independent from CO1, so they were independent markers, independent kind of types, and then we looked at them together.
So, because where I'm coming around to there is that if those eventually are going to become, I'll use scare quotes here, but standard sequences in bold, one of the things that I think a lot of people would like to see is carcinologists, taxonomists taking a look at those determinations before they go back, because there's a little bit of a circularity in that process in terms of the naming, the taxon naming of sequences that are going to go back to bold, potentially.
Well, yeah, and isn't that's the main conundrum when we do this kind of work, isn't it? And we're actually trying to, by incorporating multiple, we're trying to get around that. And in terms of who has to look at it to validate the species name, let's say, that's a question that's universal. Sure, right? Absolutely. So we... And I'm not saying that, and wow, that's the first study I've ever heard. No, but if those are going to become sort of like name standard sequences, you always want to have somebody involved in that pipeline who's not actually involved in that. Right. Well, we do have. So I don't know if you're familiar with Ricardo, but he's a traditional taxonomist on this project who doesn't do any molecular work, and he's involved in the morphological IDs.
I don't know.
Yeah, we try. Well, because we're not in BC. If we were in BC, you just would.
Okay, we're gonna thank you. So thank you.
Thank you.
And I think there were some more hands looking to ask questions, so maybe people can find you later. And I think we're going to draw this to a close just because we're sensitive to the fact that the poster people have got to be at their posters, and they've put a lot of work in them. They're over in the science complex. If you're not from Guelph and you need some directions, there are lots of signs. There are lots of Guelphites. We're basically walking out of this building, and instead of turning to the right to go to Creelman Hall and feed, we're going straight to feed in a different building while looking at posters. Thank you.